So good afternoon, everybody. Thanks all for coming to our seminar. So it's with great pleasure today that we have Dr. Nikolay Burlutskiy from Astrazeneca, and I've known Nikolay for several years already. So as Nikolay just said, I was one of his co-supervisors, and I just remember the first year of Nikolay doing his PhD when he was trying to comply with many of his supervisors' backgrounds and trying to find out what to do as a PhD thesis. And finally, he actually chose a really good topic, which was about machine learning, which I think really opened him a lot of opportunities once he graduated. So after he finished his PhD, he went to become a research scientist at Context Vision in Stockholm, and today he's part of the research and development team at Astrazeneca, where he will be presenting about the work he's doing in AI and how they apply it in oncology. So welcome, Nikolay, and thanks for giving us this great talk.
Yeah, thank you. Thank you, Nor, for introduction, and it's my pleasure to present you some of our research on artificial intelligence in oncology. So I would try to cover in the next like 40-45 minutes what is oncology and what is artificial intelligence in that context. What also we talk about digital pathology and AI, machine learning and deep learning for medical images, how it's like, what is special about it, and we touch on generalisability, interpretability, and bias in training artificial intelligence algorithms. So what is oncology and artificial intelligence? So oncology is the study of cancer, and it's the second leading cause of death globally. And what is cancer? Cancer is a group of diseases involving abnormal cell growth with the potential to invade or spread to other parts of the body. I think most of people have some understanding of what cancer is and know that it's a very leading cause of death globally, and it's 1 in 6 deaths are due to cancer. And if you look into UK, almost 400,000 cases every year and more than 166,000 deaths. And again, survival of cancer for 10 or more years is less than 50%. For artificial intelligence is intelligence demonstrated by machines, and whether like natural intelligence is demonstrated by humans. And when we talk about AI research, we mean reasoning, knowledge representation, planning, learning, natural language processing, perception, and such as computer vision. And we also can talk about general intelligence, which is a long-term goal of artificial intelligence. And again, approaches are very diverse. They include statistical methods, computational intelligence, and some traditional symbolic AI. There is a lot of discussion these days, I think, about machine learning, deep learning. So you heard about it, but let's just revisit it. So machine learning is study of computer algorithms that improve automatically, and deep learning is a subset of machine learning that uses artificial deep neural networks. So AI so far has potential to revolutionize oncology, including prevention, diagnosis, and treatment of cancer. And here is just an example of prevention, how AI actually could improve detection of cervical precancer. So in that case, like multiple screening, cervix screening methods exist, and for example, images can be taken and then AI can analyze, and these cervical cells can be collected, and then on that slide this image can be analyzed, and such methods show very high sensitivity, and it helps to prevent cancer. But I mean, in terms of diagnosis, of course, it's also important. And again, prevention is, of course, everyone hope for prevention, but if diagnosis is not that straightforward sometimes, especially for early cancers, and there are several methods have been developed where they demonstrated how AI can help doctors to diagnose, for example, lung cancer. Actually, this is paper which was written by me. So what we did actually, we had images with cancers like with tissue specimen, and we train algorithms which can automatically find whether there is cancer or no cancer in that image, and also even outline where cancer is. So a pathologist could look into the annotated area by deep learning algorithm and verify it. Of course, if you think about pharmaceutical companies, they're interested in treatment, developing new drugs, and improve the whole patient journey through treatment from diagnosis and to hopefully recovering from cancer. And AI can improve multiple aspects of cancer therapies. So in drug development, there are usually several stages. I think also many of you since due to coronavirus, I think kind of caught up a lot on what are clinical trials and what is preclinical development and all healthcare workflow. But like, let's say, if you think about drug development, usually there's a particular target identification where AI can help. For example, if you have a lot of data, then you try to cluster and see which target will be, which can be addressed with a particular drug, a particular chemical. Then you can optimize the biological process behind, like, for example, again, chemical molecule, which will will be targeting this particular like molecule, for example, like cells. And then once it's identified, then preclinical development on mice, for example, is going on. And then usually there are several, let's say, tasks which should be optimized, such as drug and dose selection, like, for example, whether it's safe to take the drug at all, or whether there is any effect, and also what is the best dose for that particular drug. And if, let's say, experimentation on mice goes well, then it goes into clinical trials on humans, and then there are phases, clinical trials, phase 1, 2, and 3. So for example, me, I work mostly in phase 1, early oncology. So it means it's usually problems of like dose escalation and seeing if like the drug has effect. And AI there is also very helpful in targeting, understanding which. Sorry, can you mute yourself? I can hear myself. Yeah, thanks. Unless you wanted to ask a question. Yeah, so actually, so in clinical trials, like especially in early phases, it's important to understand whether that group, targeted group, is responding or not responding to the drug, and it can iterate back to preclinical or even to simulations and target identifications. And then, of course, like ideally to have personalized cancer therapy, because everyone, especially with the development of molecular new generation sequencing technologies, now it's becoming cheaper and cheaper to have like whole genome, whole transcriptome sequencing, and then drugs can be personalized for particular genome. One example of such a drug, for example, anti-HER2 medicines, which can block human epidermal growth factor receptor 2, HER2 genes, making too many copies. So like cells, normal cells, they have so-called HER2 receptors, and the normal amount of HER2, I mean HER2 actually is responsible for cell growth and division, and if it's normal, then normal amount of HER2 is produced. But if when cancer starts developing in a patient, then cells, they start to grow and divide faster and in an uncontrolled way, and it can be actually these HER2 receptors can be identified using, like, for example, some molecular technologies like actually a drug can be developed which will which will actually aim at suppressing these HER2 receptors from producing too much HER2. Okay, so the second, digital pathology, artificial intelligence. So again, artificial intelligence and deep learning, as I mentioned, revolutionizing many branches of medicine, including digital pathology. So pathology is important, right? Because pathology is a branch of medicine that deals with laboratory examination of samples of body tissue for diagnostic or forensic purposes. So when we talk about oncology, pathology plays a really huge part in it, since pathologists, they are the ones who actually examine the tissue, and they can see, they're especially trained to see whether there is cancer or no cancer in under microscope in this biopsy taken from a patient. And these days, these specimen slides are scanned by high resolution scanner. So you can see this is how scanner looks like. So that a tiny slide with a very tiny tissue, which used to be examined in a microscope, now it can be scanned, and then you have a whole slide image. So something abbreviated as a WSI, which have a huge resolutions, like hundreds of thousand pixels, several gigabytes for just one image. And since it's digitized, then there are avenues open for machine learning and deep learning, which can be applied to these images. And just to give you also some background how this microscopic examination of tissue specimen is performed. So it's a routine clinical practice in pathology. So for example, if a patient has suspicion of lung or like already kind of a tumor was identified, and there is a need to diagnose that cancer, then the small that tumor is resected, and then it's sliced. You see, can resection 1, 2, 3, 4, and then a small piece of that slice is put on this slide, and then let's say scanned or analyzed under microscope. And then if it's scanned, you can see something like this. And when you slice tissue, it's colorless, so it's transparent. So that's why it's colored with special dyes, which are called hematoxylin, abbreviated as H&E. So these kind of images, you can see purple images, they're used in clinical practice, and they're called H&E slides or H&E images. There's also immunohistochemistry. So it's a specific dye which can highlight a specific structures, for example, a specific protein. So it's more specific questions to answer. And it's also routine these days, but I would say it's more expensive, and usually it's to get more information for better diagnosis. Also, like one common abbreviation in this field is formalin fixed paraffin embedded. So one tissue is sliced and taken out from tumor, then it's actually fixed, put in formalin to preserve the tissue, and then it's fixed with paraffin, so it's solid, and then it's sliced. So for example, this block could be called FFPE, formalin fixed paraffin embedded, and hematoxylin has been sliced. And then, of course, many of you, we already talked about what is whole slide image. So once that slide is scanned and you have an image, it's called whole slide image. And DNA and RNA, I think so DNA has all the information in with instruction how to produce RNA, and RNA actually produces proteins. So for example, what happens also when a tumor from a patient is taken, so like H&E slide goes to pathologist to analyze and to grade, and also a piece of tissue goes to a lab for DNA RNA analysis for gene expression, generating gene expression profile, and like DNA profile, for example, counting mutations, what kind of mutations happened there, or like methylation. So finally, like usually you have a lot of images and you have molecular data to analyze, to play with. I think most of you have computer science background or data science. So data science becomes more and more ubiquitous across various businesses and industries, including medical field. So in the past, like pathology, I imagine it was more kind of a laboratory, so based usually somewhere in the basement, where like a highly specialized like a pathologist would deal with microscopes and tissues. Now this is scanned, tissues are scanned, and data scientists, they work on intersection of software engineering, math, statistics, algorithm development, and data communication, and work with large data sets, these images, and developing pipelines to processing the data in medical field, like this whole genome, whole exome data, and H&E images. And then, the question is, you need to communicate these results usually to domain experts such as pathologists. So what very often happens, for example, a pathologist says, IHC stained primary tumor specimen demonstrated highly overexpressed HER2 protein. So if you are not in this field, then you think what you're talking about it. But I mean, it's, pathologist way. This is the way we speak. But if you think about deep learning expert or machine learning expert, they would say something fully convolutional neural network, deep lab version 3, reached F1 score of 0.82 on the test set. So pathologist is also, for pathologists, it's hard to understand what is it. So, I mean, a lot of work is happening, I think, to speak the same language, and at least with Astrazeneca, I found that it's happening, and we are very interdisciplinary, and I believe it's not only within like industry, but of course academia and stuff like that. And there are already several examples when pathologists and data science fruitfully collaborating on many impactful projects. So here an example, also some work which I worked on. So this is how H&E image looks like, and the tissue comes from prostate tissue specimen. And then we train an algorithm with annotations from pathologists. The goal was to train an algorithm which can find cancer, or say it's benign, and not only find cancer in the image, but also outline it. So for example, this on the right, you can see that it's the model which we trained. It could accurately outline a tiny gland which was cancerous. And then we asked pathologists to annotate, and you can see this pathologist annotation deep learning model, they correspond very well. And again, that image wasn't part of training set, so the model didn't see this image before, so it was test set. So it's very exciting how deep learning actually can help pathologists not to miss small areas of malignancy, because again, whole slide images, they're huge, so it takes a lot of time and effort to go through all these slides. And also potentially can provide survival rate estimates for a patient and suggest optimal treatment, predict cancer stage automatically. So there are many tasks which pathologists are very excited to be solved with machine learning, deep learning. Yeah, so here we come with what is actually machine learning and deep learning, and what kind of methods we use in training such algorithms. So as you know, some of you may know, at least I'm sure you heard, that machine learning solves different tasks, such as, it could be segmentation, classification, detection, regression. So again, just some definition. What is segmentation? Yeah, when, for example, you have an image and there's a cat and you want to outline. So the yellow color, it's semantic segmentation of a cat, and it's also segments grass, cat, tree, and sky. So it's multi-class semantic segmentation. Yeah, we have 4 classes. And the output of segmentation could be segmentation, like outline, segmentation mask, or probabilistic map. So every pixel is assigned with a probability of being like grass, cat, tree, sky. Then can be classification and localization. So classification, we know that there is a cat in this image. So we don't know where exactly, but for classification, we know that there is a cat in this image. And for localization, we have a bounding box, so we know where the cat is. So it would be called classification and localization. And it can be a single object. It can be multiple objects in the same image. For example, for object detection, we can detect 3 instances. Yeah, of 3. There are 2 dogs and 1 cat. And for instance segmentation, so we see that there are 2 dogs and 1 cat. If, for example, it was just semantic segmentation, these 2 dogs would have the same color. And for example, semantic segmentation tasks are very common medical field. And if you think about it, I just put like quite randomly like different images, but for example, in brain segmentation of brain parts, and again, it's also can be three-dimensional segmentation. It can be like, again, CT scan, lung nodule segmentation. It can be ultrasound, also segmentation, or whole slide images you can see here. It's a whole slide image with different probabilities assigned that it's cancer or non-cancer. And again, segmentation can be used for, it's very common task for medical images. And usually state of the art, the natural process, what you do in machine learning, you define data for training, validation, and then you tune parameters on validation data, and then you have a test set to report the performance. So yeah, it's again for training models, again, very different algorithms which you usually would try, like logistic regression, decision trees, KNN, and for validation, you can like find optimal parameters for your model, and then you can train model on all your data, and then you would like to have a test set which is quite representative for the distribution you want to predict for. So like, for example, if it's a population of images coming from potentially cancerous patients, then you can evaluate on independent test set and then report the performance. And when you report the performance, again, depends on task, but quite common. Let's say if it's a classification or segmentation, you deal with confusion matrix when if you can have true positive or true negative, right? So it's like you have, for example, binary classification, cat, no cat. So if it's predicted as cat and it's cat, it's true positive. If it's not cat and it's predicted as no cat, true negative. And then you have false negative and false positive. Yeah, so you have 4 numbers, and then you can combine these 4 numbers, and then you can get precision, recall. You can combine precision and record and have, for example, F1 score. So it's and then you can plot like area under a curve. So there are many ways how you can report the accuracy. Deep learning, I think also many of you heard quite a lot these days. So deep learning works, to simplify it, to make it simple, works the best with large data sets solving nonlinear problems. And it's especially showed its great performance on images and text, problems involving text. So if you think also conceptually, classical machine learning usually requires feature extraction step, but can be avoided using computationally extensive deep learning. So again, back to cats and dogs. Like in traditional machine learning, you would do some feature extraction, maybe extract like, features like nose, ears, or like some body of the cat, and then combine them and run some classification model, and then the output can be cat no cat or car no car in this case. So in this case, maybe you need to extract like wheels, roof, the shape. But for deep learning, the beauty of deep learning, you don't need to do feature extraction because the model itself can convolutional filters can run through the whole image and generate many features which representative, which you will eventually will be, if a model trained and performs well, then it will be like some features related to, so some layers will learn that, some feature layers, feature maps will learn that like these are wheels with a roof and stuff like that. And deep learning attract a lot of attention, and it's dominating leaderboards in machine learning competitions and challenges. So how it started, for a few challenges, for example, 2012 challenge, but I think it ran even before, like imaging challenge, where anyone could train their model and predict on a test set. There were 20 classes, 20 different classes with 12,000 different images and 27,000 regions of interest and 7,000 segmentation. And then classic machine learning approaches, they had performance, but once deep learning methods showed up in 2012, then since then, all these leaderboards are dominated by deep learning approaches, which I think shows that they're superior in at least segmentation, classification, working with images. And even more, like there was a challenge with 1,000 classes. For traditional machine learning, when the challenge was with 20 classes, 1,000 classes sounded like a crazy task. Since AlexNet, the challenge like 1,000 classes doesn't sound that crazy, and that dataset like ImageNet is now used for pre-training models, which then can be used for fine-tuning and reusing on your custom dataset. And I think the latest challenge, the accuracy of the model was quite comparable with a human annotator. So if you have a test set and you ask a human what is in the image and choose one of like these 1,000 classes, then deep learning approach and human annotator, they perform quite comparably. But again, segmenting classifying images was already a mature field making before 2012 or so, and before deep learning, and so like it makes deep learning breakthrough even more impressive. So here I think I took some slide. I was preparing some presentation for more like classic algorithms for example segmentation. So we've been this research is quite old, and there have been developed many methods. So, but now since deep learning is there, I think a lot of these papers are, at least at the moment, less interesting, less exciting. But of course, always nice to revisit and keep them in mind. So deep learning actually and deep neural networks, they are all based on a, I mean, you can think as the like simplest element could be perceptron, which I would call it the grandfather of model deep neural networks, and it was invented in 1958. And the idea is pretty simple. So you have inputs. In case of images, it can be pixels. Every pixel has RGB value, like red, green, blue. So it's a value from, if it's 8-bit, from 0 to 255, and then you multiply these pixel values on some weights, which actually I learned, and then you sum up, then you apply some activation function, some nonlinearity, and then you get your output, which, for example, is a value which you can surrogate probability of that image being a cat or not. So, for example, from 0 to 1. And this, I mean, when I think about deep learning or like deep neural networks, I think this is a building block of an algorithm for supervised learning of binary classifiers. Of course, there are like since then been a lot of improvements and architectures developed for again image classification and segmentation. So for classification, the basic model is so-called convolutional neural networks. So it's neural networks which has convolutions as basic operators. And convolutional network, to simplify, again, it has an embedder. So first part, where like image is your input, and then there are several layers. Yeah, so that's why it's called deep. And then you run convolution and you reduce the dimension of your image, but then you get like feature maps, which represents some features, for example, for cat, like eyes, nose, and so on. And then once you go down to reasonable number of features, which represent on a high level, what is abstract representation of a cat, for example, then you have classification part, which is fully connected layer. So it's which actually, when you go like, for example, from 1,000 features to one feature, which is one output probability of that image being a cat or not. But I mean, for segmentation, you need to classify, like, let's say, every single pixels, and what you what happens actually, you take the first part, the embedding part, embedder, you throw away fully convolutional part, and then actually you have some features in here, which is say 1,000 features, whatever. So it's a very tiny image, and then you deconvolve or actually go back. So it's a decoding part, and actually you go back to the same size of image, which actually will be like, for example, for cat classification, like only one class, right? Cat, no cat, and then you have outline of a cat. And then, of course, you have these features from bottleneck, and also you have information flowing from different layers because you lost a lot of information, right? So resolution went down. So this is like the general idea of fully convolutional networks for segmentation. And yeah, for convolutional networks, various convolutions, deconvolutions are building blocks. And yeah, convolution, you can think about it as a mathematical operation. It has a kernel which runs through the image like a window, and then through like multiplication and sums, it's converts, let's say, your image to feature maps, which then go through again for convolution and so on. And then transpose convolution that way when you, for example, go back. So finally, again, for fully convolutional networks, you go down. So you kind of encode your image, and then you need to go back because you want to reconstruct the segmentation mask of the same size as your input image. So for that, you have transpose convolutions, and of course, there are many smart ways how you can deal with like limitations of how, I mean, you have to run it. It's a very computationally expensive task, so you run it on GPU, and running on GPU still have limitations of memory. So there are many efficient ways how you can run like quite large images on GPU without losing much context. Because again, for example, for full slide images, you cannot put like 100,000 by 100,000 pixels image in GPU. So you have to split it in smaller patches, but then you need to reconstruct the context. So for segmentation tasks, UNet, so-called UNet network, is one of the most popular implementation of fully convolutional networks. So if you read papers on especially medical imaging segmentation tasks, UNet would be quite often the model to start with as a baseline, or to compare against. And as you can see, again, you have input image, you have a few convolutions, then you have max pooling layer, and then again you go down, you have a bottleneck, and then you go up with this deconvolutions, or like transposed to be more accurate convolutions. And then you have also so-called skip connections, when actually you copy and paste like part of feature maps from previous layers. Since like in bottleneck, you lost quite a lot of context information. So yeah, so there are these days, I think, quite a few resources to find state-of-the-art architectures and learn more about the latest deep learning research. So I think you're familiar with Archive, where you can find papers even before they get published. I think it's very fast-paced environment, a very fast-paced field. So Archive, I think, is very useful to get papers open review. Like, yeah, these days I think it's also a lot of code is shared on GitHub, and there is also a nice resource called Papers with Code, where you can find state-of-the-art algorithms for, again, segmentation, classification, and so on. So yeah, so deep learning and medical images. So also there are some specifics working with medical images, and also just to tell you, this is, I think, one of very exciting project which was part of Kaggle. So there is a website called Kaggle, where it's a platform where anyone can join, and so it's like machine learning problems as hosted there. So anyone can join, try to solve it, and if they solve, sometimes they can even win some money, and it's hosted by companies or some more kind of government organizations, or like academic organizations, and sometimes it's just for knowledge sharing. But I think this one is interesting to mention. Data Science Bowl in 2007, which was a challenge of improving lung cancer detection, and the prize money was like $1 million. And the problem was stated as using data set of thousands of high-resolution lung scans provided by the National Cancer Institute of the United States. Participants developed algorithms that accurately determined when lesions in the lungs are cancerous or not. So this is a sample of MRI scan, and then you need to find some lesions and whether they're cancerous or not. So you see, like, I think this kind of, I think prize money also show how much interest in such tasks of identifying cancerous areas automatically and applying AI to imaging, medical imaging. There's also so-called grand medical challenges, which try development of deep learning methods, and it's also a website. You can find it on grandchallenge.org, where it's like a Kaggle, also like different machine learning, deep learning problems are hosted, and then everyone can participate, and then usually where images provided, and then you can train, and then usually once an algorithm is trained, it could be part of publications. So many medical conferences these days, they organize, for example, MICAI conference, they organize these challenges. And a lot of data is there with annotations. Yeah, so for example, one of example was, now I think you all know what is HER2 and H&E. So one challenge was to score HER2 H&E images. It was last year challenge. So again, you are provided with H&E images, and then on a slide level, you had annotation like score, HER2 score is from 0 to 3. So it's like 0, 1, 2, 3. It depends on how HER2 is expressed in the image. So HER2 0, no, like it's normal. And 1 is expressed, overexpressed, and HER3 is overexpressed quite a lot. So actually it's quite a continuous problem, but there is a guideline for pathologists how to grade it from 0 to 3. And there was a challenge to organized to train deep learning algorithm to score this. Also, nuclei segmentation and classification is very important task, because again, morphology of slide specimen affect like how a person, let's say, you can see how a person responds to drug, or whether that person will respond or not, how immune system responds. And again, you can see also different morphological features, different types of type of tissue and cells. You can see lymphocytes, you can see macrophages, neutrophils, and for example, one task was on this challenge was multi-organ nuclei segmentation. So you're given H&E stained tissue images of 4 different organs, and these cells, they look quite differently in different, potentially can look quite differently in different organs, at least like orchestration of this like tissue and cells. And participants were used this annotated dataset to develop computer vision algorithms to recognize these cell types from tissue images. Yeah, so this is our tasks, and for example, when you work in the medical field as an AI scientist, so you develop deep learning applications and applying state-of-the-art techniques to various cancers and problems. For example, if you talked about whole slide images, they're huge, and you have different cancer types. It can be lung cancer, prostate cancer, breast cancer, and different types of staining. As I mentioned, H&E, hematoxylin and eosin stained image, the purple image. It can be immunohistochemistry image. It can be multiplex immunofluorescence. It can be computerized tomography, CT, MRI, whatever. So we're quite plenty of modalities in medical field. And tissue it can be tiny biopsy, it can be resection, it can be a huge piece of gross section of tissue, and then potentially would like to segment out cancer to classify, find regression. For regression, for example, you have a whole slide image and you want to run some survival analysis. You want to predict, let's say, how long that person potentially will survive from now on, and or you want to cluster. Cancer is very heterogeneous. You want to cluster patients in the groups. And also we can use different types of machine learning approaches, like supervised, unsupervised, semi and weakly supervised approaches, which I'll touch upon. And yeah, also mentioned that very often in the real world applications, you employ several deep learning models at once. I took this example from a paper, just simple example where you have two different deep learning models. One is for tissue segmentation. So you can see again this CNN, so convolutional. Like actually it's fully convolutional network. You see first it's convolution max pooling, so it's encoding part. Then you have convolution part, so it's like bottleneck. When you can see your feature maps are quite small, but there are many of them. And then you go up with the deconvolution, and you end up with a tissue outline. So you know that black one is background and everything else is tissue. But also, like, you know, once you know where tissue, you would like potentially have some cell detection. And in this case, you see you have single cell detection classification. So again, you have convolution layers, and then you have fully connected layers. And then actually as a output, you would have some detection. So you have, let's say, X and Y. So you know coordinates in this whole slide image where a cell is, and not only where a cell is, but also class of the cell. So it's like classification plus detection. This is also like quite often what happens when a patient, a biopsy or like a resection is taken from biopsy. It's important to understand whether a patient is how immune system of that person responds to a drug, to treatment. And there is a definition of like immune cold or immune hot. So immune hot actually when you have many immune infiltrates within tumor microenvironment. So it's like a tumor. It's called tumor microenvironment. So there is like a quite, at least for me, who is not a pathologist, quite complicated combination of different types of cells. And when immune cells infiltrate tumor microenvironment, then it means that immune system responds and fights against cancer. And even in the image, you can see that it's so called so immune hot. And I mean, you can see like there is difference in these two images. And pathologists also can see immune hot, immune cold areas, and it's possible. I mean, in a recent paper, it was shown that AI can identify immune hot and cold regions and H&E images automatically, save pathologist time. Yeah, the main problem with medical images and medical data in general, it's scarce, very noisy, and labels are hard to obtain. Scarce, of course, it's many regulations over images. So it's not just go outside and take pictures of cats. You need consent forms from patients. And again, you need to have patients which are selected, for example, for a particular group of particular cancer and with particular like demographics and particular characteristics of like cancer type. And labels, I mean, for example, I cannot provide labels where cancer or not. We need the trained, highly specialized pathologists, domain experts, and it's very expensive. So I think I convinced you that it's a problem. And yeah, so when we have annotations, so in cases when, let's say, we have cats and dogs, and we have someone classified already, this is a cat, this is a dog, then supervised learning can be applied. And it's very powerful when there are labels for samples. But again, when we don't have labels, we can use unsupervised learning. And for example, for supervised learning, it can be classification. I like this example, divide the stocks by color. Or it can be regression, divide the ties by length. And for unsupervised learning, we don't have labels, so we can cluster divide by similarity. So we apply algorithm which try to kind of automatically, you know, according to define the similarity to cluster these samples. Or for example, sometimes we want to run some dimensional reduction. So for example, if you have 20,000 genes, maybe we are not interested in all of them. We want to reduce only to the genes which drive cancer mutations, for example, and then we can run some dimension reduction. And again, as I mentioned, training an algorithm is supervised learning settings requires labels what will serve as the so-called ground truth. I like this name. So the annotations, they're called the ground truth. So for example, when we ask a pathologist or annotate where's cancer, where's no cancer, it's ground truth for us. And yeah, these annotations can be provided by domain experts, and usually also it's very important to have a well-defined annotation guide, especially in cancer, because it's very I mean, the same what is tumor can be defined very differently. What you include? Do you include only tissue, or you include stroma, or like, you know, what happens if, like, let's say if it's as I showed, like, you know, when tumor microenvironment is invaded by let's say immune tumor infiltrated lymphocytes. It can be included, it can be not, or necrosis, for example. So in most of cases, whatever we have, these labels are noisy and ambiguous. And in that case, semi-supervised learning and weakly supervised learning can be powerful. So in that case, you have, for example, annotations only on slide level. I know in this slide, there's cancer. Where? I have no idea. Then you can apply weakly supervised learning and try to find areas, or like, let's say, pixels or small tiles patches where potentially there's cancer. So then it can be provided some sort of heat map, and these cancerous regions detected by cancer deep learning actually can be sometimes very accurate. Also, again, when we have a lot of scarce images, we would like to actually get some data from the data we want to apply transfer learning. Yeah, so transfer learning helps us when data is scarce, and it can allow us to save computational effort and time. And again, so for example, if we learn the task, then we can reuse that model and fine tune on the data which we have. And this is also an example which actually I did quite recently. So we had annotations only on whole slide image, and we managed to train an algorithm with noisy annotations, and also we apply some transfer learning from immunohistochemistry. You see these brownish images. We train an algorithm on HC images, and then we transfer it into H&E images. So you can see they're different. It's same. It's not the same tissue, but it's adjacent tissue. So morphologically, it's very similar. So it's possible to transfer. And of course, when many people say that deep learning is a black box, so and it doesn't generalize. Usually, you train on dataset A, and then you go to dataset B, and the problem is how generalizable it is, and interpretability and bias. So to achieve generalizability of trained algorithms is challenging, but of course it's a task. It's our ultimate goal. And again, there can be many different reasons why it happens. Changes in a pattern over time. I mean, differences practice between health systems, how the tissue is sliced, prepared, stained, demographic variation, patient genotypes, hardware and software, how it's scanned, variations in disease, and so on. Then interpretability of trained algorithm, it's something also active area where when we learn something, we want to actually ideally have an interpretable algorithm, which is trustworthy. And it means it's potentially reveals the underlying rationale of a model making decisions. So just to give a simple example, if we train a classifier, whether it's a bird or not, would like, let's say heat map or like a probabilistic map where the model is taking information from to be around the bird, not about, let's say, the branch it's sitting on or some, grass. So something the same is, if we talk about cancer, we want, let's say, information be taken from some cancer regions, for example. And of course, we would ideally like to have some models which can explain themselves what they learned and how they learned, and so we can trust more. And again, it's we can also validate deep learning approaches using biological methods. So here an example also from one paper. So you train algorithms to detect different types of cells, but then you can take the tissue and stain with a IHC staining, which actually highlights a particular types of cells, and then using these chemical annotations, which provide some biological ground truth, you can validate your algorithm, which was trained only on, for example, H&E images. So it's also an active field of research. And also, once you train an algorithm and you're happy with its generalizability, maybe it's even interpretable, Then, of course, we would like to know more about potential sources of unwanted consequences and biases in that algorithm. And again, when you train your algorithm, let's say, on data from patients, there are many biases, like representation bias, for example, how you select your population. So, I mean, cancer patients, so already representation bias, but of course, we're interested in predicting cancer, no cancer. With historical bias, yeah, how we generated the data, how many legacy data sets, then how we measure, how we capture that images, like let's say brightness of pixels, bright field, camera characteristics, how we define our model, how we evaluate. So, I mean, there are plenty of biases to keep in mind. So I would say that building trustworthy and ethical AI is crucial. I know that we're not part of EU anymore, but there have been some work on ethics guidelines for trustworthy AI, and it had 7 requirements, key requirements, such as human agency and oversight, technical robustness, safety, privacy, data governance, transparency. So it's a quite large document. So if you're interested, you can have a look. But I think like there's a lot of work, going on to make sure that we build trustworthy and ethical algorithms, especially if they go into some support, even support of making some decisions, crucial decisions, and suggesting, like let's say, diagnosis or treatment. So here I would like to recap some research questions. So this is kind of research questions we usually face. Yeah, is it possible to train models to distinguish cancer types, for example? For example, squamous cancer, or squamous lung cancer, or adenocarcinoma. Then also, what does a model learn from images? So we'd like to kind of also interpret and see if there's a biology inside. So in that case, like you want to somehow extract that information and show it to domain experts. Of course, like since data is scarce, we would like to transfer learned knowledge into another domain. We learned on breast cancer, we would like to transfer it to lung cancer. And we would like to automate clinical workflow in many stages. And again, impact, I think it's a lot of impact there. Faster and more accurate diagnosis is AI can help with it. Discover new patterns, biomarkers, new drugs. We learn more about cancer, we learn more about potential biomarkers, and we develop more drugs. And of course, we also want to be agile. And I would say this such project socially rewarding and motivating. So I hope you also will get more involved in such projects, unless you're already working on. Yeah, I think that's it. That's all my presentation.
Thank you, Nikolay. You really gave us a really big overview of what you're doing, and there are many aspects of applying the different algorithms, to the ethical aspects, to actually how to involve as well experts in this area. Does anyone have any question for Nikolay?
I can see there's one question in the chat, so I can address it. So in deep learning, are the feature extraction dynamical? So I would say it's what happens. Let's say you have as your input, for example, image, and you have some ground truth, which is cancer or no cancer. And then what you're trying to do, like imagine this is your input, your pixels, and then you want to train an algorithm which says it's cancer or no cancer, but you know truth, which is, let's say, cancer. And then we train these weights, so we randomly initialize these weights. We learn, so we get the answer, and then it's before we, and then we calculate the error, which is the difference between ground truth and the answer by the algorithm. And then usually the error is not 0, so you try to iterate it again. So you kind of use backpropagation to change the weights, and then you iterate. So you think it's like optimization problem where you want to minimize the error, which is done over time. And of course, like at some point you'll have convergence because maybe your labels are noisy, maybe the model cannot learn, or something else. And at that point you stop and all your weights are frozen. So then deep learning model doesn't learn anymore, and then use these fixed weights to predict. So once you have a new image, you just run through these weights, like multiply, sum, and get your answer. But I mean, of course, like there is continual learning when, for example, you train that model and more data is coming, annotated data, then you can continuously learn, and or a new data set is coming, or like it's a continuous stream of images. So potentially it can be done. Of course, there are many regulation problems in medical fields. Usually, like if you train this algorithm, it goes through some approval, whether it's like European, like CE marking, or FDA. In the States, and it's quite rigorous in the sense that most of the time you have to fix this algorithm and don't retrain. And if you want to retrain, improve it, then you retrain it, but then you have to go again for all these approvals.
There's a question. There's some questions on the chat as well. So I don't know if you can read Federico's question. Given the need for large training data sets with ground truth, with deep learning, what's the state of the art for few-shot learning, semi-supervised methods, etc., in relation to oncology?
Yeah, so I think few-shot learning is, of course, it's very interesting because I mean the problem is we don't have many annotations, but potentially we can have a lot of images. I mean, frankly speaking, I haven't worked much on few-shot learning, but I mean for semi-supervised learning, of course, there's a lot of interest when you have annotations, images annotations, like just a subset, and you include your images without annotations and try to learn and see if performance of your algorithm improves. But then, once you train these models on some particular tasks, and usually in some supervised or semi-supervised way, then you can use that trained model for transfer learning, applying like few-shot learning. So you take this large model and then you try to fine-tune it on just a few examples, and or just go no shot.
So Akram continued as well in the discussion. He's talking about the biases. So how that's very hard, almost impossible to work out when there's biases. So how do you deal with that?
I would say there are always some biases we know. There are some biases we have no control of, right? So we know some of such biases. And I mean, like when you work in oncology, most of your patients, they come from patients who had cancer, and in that case, it's one bias, right, potentially. But I mean, we know about this bias. Sometimes we have bias which you have control on. So for example, if we scan with one scanner, like a particular image, and we know there is bias to that particular scanner, then we can scan with another scanner, so we can deal with that. So some biases we can address. And also there are some biases which we don't know, and of course we can learn. So for example, we can have a hypothesis what is a particular bias there, and but of course we will never be able to cover all of them. I think the idea is like try to understand what are the most kind of important biases and which we can address to some extent.
Kathy, you have your hand raised. Do you want to ask a question? You can unmute yourself.
Thanks, Noor. I do. Nikolay, you mentioned interpretability, and you said that was an important issue, and I would agree with that. But my understanding of the current state of things is that we don't have interpretable deep learning models. Am I wrong about this? And if there are some, where do they exist? What do they do?
So you can think about from different sides. So one thing is, imagine you have end-to-end deep learning model, which given a whole slide image, it tells cancer, no cancer, and no explanation. Yeah, no interpretability. Then next level, it doesn't tell you that it's only cancer, no cancer. It also tells you that algorithm decided that it's cancer because this is tumor area, this is cells, these are, let's say, immune cells, these are T cells, B cells, and they're concentrated in this area. That's why it's cancer. So it's better. So it's already gray box. Yeah, and the pathologist can confirm. So pathologists can build trust in the algorithm because the algorithm actually identifies where cancer is, the cells, some morphological let's say primitives, and then based on these features, which are confirmed by pathologists that they are working well, decides if it's cancer or not. Then, of course, if you look only where cancer, where not, then yeah, it can be a black box. But there are several methods which you can look into, for example, GradCam methods where you can try to build a map, like a heat map on so how let's say your model makes decisions on which kind of neurons, neural networks are more important, and also let's say some methods to rank importance, like SHAP or LIME method. So I think it's quite active research, and again you can use some occlusion maps. So you take your, let's say, H&E image and you just put a black box where cancer area is, and then of course you would expect the model to say it's no cancer anymore because you covered. So I mean, you can run some of these kind of experiments, or some adversarial experiments. So yeah, I think it's an active area, but I think it's let's call it gray box at the moment from my perspective. And of course we would like to learn more about how to interpret the models.
Okay, thank you.
Can I ask a bit of a detour about potential in material science?
Mm-hmm.
It's actually because I work in a department where we one of our ongoing tasks is actually looking at filters and various other things for hazardous materials.
Mm-hmm.
And one of the questions was going to be, is this mostly, because I'm not an expert in AI or the biology side of it, is it mostly analyzing the image once you've got the image, or is it also actually having something that operates the microscope and finds the image and is able to also focus and actually scan a set area and then actually find the image itself, and then potentially then characterize using the morphology that it sees from the image? We're also looking at compositional analysis, so it's actually electron microscopy. And yeah, we're really interested at the moment in whether or not certain potentially hazardous fibers or other things could be identified and counted automatically by a system, because in this case it's mainly just quite a laborious task for an individual to have to flick from screen to screen counting a certain area. So yeah, I was wondering, I think you're clearly mostly focused on the biology side of things. I've just quickly looked up material science and deep learning, but I'm wondering if you know anything about that potential side of it, because you even talked about prevention is the ideal thing, and this is even part of that. If we can improve and speed up the analysis and find out that there are either hazardous materials or even the ones that accumulate individually, it's not like they're toxic or poisonous, but even with potentially microplastics in an FTIR, I don't know if I need to explain that further, but using the spectrum and what you see in the microscope to actually automatically count things, that would take scientists hours or days to
Yeah, I think the task which you described, for me, it sounds very similar to what pathologists also do, because imagine it's a tiny tissue. It's a few millimeters long and very tiny. But I mean, when you look on the microscope, it's like so many like cells. It's in terms of pixels, like a lot. And so many pathologist has to count. So choose an area, a region of interest, and count how many cells are positive or negative. And deep learning is very good in identifying like positive, negative cells. So this is definitely one area which I think what you described can be also very beneficial. So to identify like regions of interest and do some counting, because we know that computers are much better in counting than humans, right?
Yep.
And so it's something pathologists are already doing, and there are quite many for this automatic quantification of regions and images. I think it's, and in terms of algorithms, I think it's very transferable. The same approaches can be done, and again, like to identify some areas of potential hazard or some kind of pathologist, trying to find, again, it's like trying to identify, let's say, imagine you have a huge display and only one pixel is broken, and you want to find it. If it's a different color, maybe you can identify this easily, but again, like machine learning, deep learning can do it much better than humans. And again, identify cancer. Imagine you have hundreds of thousands by hundreds of thousands pixels, and then there is a tiny gland which is potentially like, I don't know, 20 by 20 pixels. So a pathologist has to go through all of this. So I think like in your area, definitely, the same methods, I would say, could be applied. Maybe there is a requirement for doing it faster. And again, back to like more kind of hardware part. So Google, they developed, they call it virtual microscope. So the idea is pathologists, they look into microscope, but what they see, it's already like, let's call it augmented reality, right? So the algorithm behind working and identifies where cancer was not, and then you can see the whole slide image. The slide itself. So it's still kind of very hardware based, but then you can see which areas cancerous or like suspicious. And of course, the requirement would be these algorithms must be optimized, because computationally they're very expensive, and in our case, we can wait. In most of cases, like, when slide is taken, patients can, at least at the moment, we can wait like several days. Of course, ideally to have it directly, so to make it faster. But the current practice, pathologists get cases and they are overloaded with many cases. It can take a week. And, patients, they're, earlier they get some treatment, the better. So definitely will help.
Yeah, thanks for that. Because I was just going to say for us, when we're trying to identify, first of all, that it is a respirable fiber, that it is of a certain composition, and that it is potentially hazardous. You can get so many fibers which are completely safe. It's almost like, I think, a needle in a haystack.
Yeah.
Doesn't quite cut it. It's like a needle in a stack of other needles that are a slightly different composition. So with what you're saying about trying to identify as well as not ignoring, but very quickly spotting which ones aren't of a concern. It's something we're definitely looking to pursue. So that's why I came here, to try and learn. I think looking at it would be a probably supervised learning type of thing. It is a job where the actual analysts themselves, because it's also regulated quite heavily, the analysts obviously have to know what they're doing. It's just a case of speeding up and automating certain things, because I wasn't sure if some of the things you were saying, that the AI can even sometimes be better than the actual doctor.
It's hard to say, because like usually we are trained algorithm on their ground truth, right? So we if it's supervised learning, we train. So our ground truth is from pathologists, and if you also ask pathologists, they're all trained, and there is a lot of variability there. So it's also, but it's very hard, and I think it's also very, how to say, not great because I think pathologists also potentially can get like, threatened that, AI is going to replace us. I mean, it's not, I think, they can.
Yeah, I was going to
it's
admit
help,
that
not to replace, because again, counting pixels, counting cells, it can take a long time, and it's better maybe to do some research and concentrate on complicated cases than to spend 80% of your time on like some easy cases to count cells. But yeah, I mean, I think when it's hard to define what is better, right, than pathologists. So I mean, usually you compare like some variability and calculate some kappa score, like try to see like a concordance between different pathologists and algorithm, and if it's kind of sits somewhere in between, then it's great.
Well, I guess the big thing at the moment is that the computers are only going to get better now.
Yeah.
So even though I don't think they're as good at the moment, they're definitely gonna. Yeah, I may be working towards making me and my colleagues redundant slightly, but yeah, someone's gonna do it. So, but thanks for that.
I think you still have some work to do to train the models and label them. So
hopefully,
yes. Okay. Thank you for the discussion. I think we have a set of questions, and thank you, Nikolay, for answering them all and for your interaction. So I think everybody has welcomed your talk, and it was quite enjoyable. I think just giving us a quite overview of everything you do is really great. I think we can ask many questions, but you gave us a really great overview. So thank you all for attending, and hope to see you soon in another talk. We're gonna